I'm going to try not to just completely skip much. But when we get to slides that we've already covered, I'm probably going to just kind of briefly mention what's on there as a reminder and move on. But everything on those slides is fair game for a quiz or an exam. It's just we've already done the lecture for those slides. We don't really have time to go over them completely again, and you probably get bored listening to them over again, because you already know them. So the nervous system is one of two organ systems in our body that helps to maintain communication throughout our body, and that communication is going to be used to maintain homeostasis. So remember, homeostasis is trying to keep the conditions in our body more or less the same over time. And so the endocrine system and the nervous system are what's going to control that. The endocrine system, remember, communicates by chemical signals, and the nervous system is going to use a combination of electrical signals and chemical signals to send messages. The chemical signals are more or less going to be kind of in the synapse, very, very close communication from one cell to another, whereas the endocrine system uses the chemical messengers over long distances. They just put them into the lymphatic system, they put them into the bloodstream, and it travels throughout our body and can relay that message to a lot of different things. What the nervous system does to maintain this communication has three main parts to it. Number one, there's a lot of words here, but it basically boils down to things on the periphery of our body. Our appendages, our skin on the outside are going to receive signals. They're going to sense the external environment around us, and they're going to send signals to the central nervous system, meaning the brain and the spinal cord. So things on the outside of our body are going to sense the outside world and send the information to the central nervous system. Central nervous system is going to receive those signals. It's going to more or less think about what needs to be done, and then determine and make a choice about what the body needs to do. And then it's going to send signals back out into the body, whether it be an organ or a gland or a muscle, or whatever needs to be done. It's going to send those signals out in order to have them acted out, so that we can interact with the external environment that we sense in the first part. So the nervous system has a whole bunch of different subdivisions. We're not going to talk in depth about the divisions too much today. There's a few slides about them, but we'll talk more in depth about them in the next two lectures. We talk about the central nervous system, the peripheral nervous system, and then the autonomic nervous system, which is going to be also include the sympathetic and parasympathetic divisions. So we're going to start up here, and it can be broken down, and then each subdivision can be broken down further and further and further. So when we talk about the central nervous system, it is the brain and the spinal cord. If something is happening that is not happening in the brain or the spinal cord, that is going to be the peripheral nervous system. Think about the periphery of our body. That's going to be the PNS, and the brain and the spinal cord is the CNS. So the peripheral nervous system, we said, is everything except the brain and spinal cord. It's going to be made up of nerves and nerve ganglia. A nerve is going to be a bundle of nerve fibers. A nerve fiber is an axon. So take a bunch of those nerve fibers, bundle them together, wrap them in connective tissue, and that's your nerve. A nerve ganglion is going to be a nerve, and then essentially you're going to have multiple nerves coming together, and they kind of get wrapped around each other in a knot. So it's like a bulge. It's where the different nerves come together to talk to each other. The peripheral nervous system, like we said, is going to be taking in information. This is going to be part of the sensory division. Sensory can also be called afferent with an A. This is going to take signals from the receptors in the periphery of our body. It's going to send them to our central nervous system. The sensory division has the somatic sensory division and the visceral sensory division. Somatic sensory division is going to be things like our skin and our muscles and our bones, not what you would think of as our major internal organs. The visceral sensory division is going to be coming from those internal organs, things like our heart, stomach, and bladder. You know when your bladder needs to be emptied. That's coming through the visceral sensory division. The motor, or efferent, so this is afferent with an A, this is efferent with an E, is going to take signals the opposite direction. These are going to be the signals coming from the central nervous system out to the periphery of our body, the glands and the muscles that are then going to carry out those instructions. The somatic motor division is going to be signals from the central nervous system to our muscles. They tell our muscles when to contract. The visceral motor division is going to be part of the autonomic nervous system that we'll have a whole lecture about. It's going to carry signals to glands, internal organs, cardiac, and smooth muscle. So this is skeletal muscle, cardiac and smooth muscle, but similar between those two, but different than skeletal muscle.
Involuntary.
They're involuntary, right. So the somatic motor division is going to control the voluntary skeletal muscle. Visceral motor division is going to affect everything else that's involuntary. The visceral motor division also is going to have something called the sympathetic division and a parasympathetic division. And again, we'll go more in depth about this in the autonomic nervous system lecture. But essentially, the sympathetic division is going to be sort of your fight or flight mechanism. This is what's going to turn your body on. It's going to make you more active, ready to fight or to run. It's going to make your heart beat faster. It's going to make you breathe faster, and it's going to stop digestive and urinary system. Now, if you need to fight or flight, this makes sense. Increasing your heart rate, and increasing how fast you're breathing. That makes sense. Why would you want to stop the digestive and urinary system? Right. If these are working, you're using energy in blood supply to control them. If you know you're going to have to fight or run for your life, you want that blood in your muscles to make them work as fast as possible. Generally, working in the opposite direction is the parasympathetic division. This calms things down. This turns down your heart rate and your breathing, and it's going to turn on the digestive and the urinary systems. Usually, most organs are going to actually be receiving signals from both of these at the same time. And so it's going to be a matter of which one is stronger at a given time. If this is stronger, we'll have the fight or flight response. When this dies back down, this kind of takes over. This is more constant. This goes up and down. Okay, so that is just a very, very brief overview of the nervous system. Now we can talk about the nervous tissue itself and the neuron. And so there are some properties that all neurons are going to have in common, and these are things that we've talked about. So number one is just the ability to be excited. So we've had two different excitable tissues. There was nervous tissue, and what was the other one? Say it again? The thing you just took an example. Muscles. Muscle and nervous tissue are both excitable. Neurons are also going to be able to conduct electrical signals. So they're going to receive signals, transduce them down their cell body, through their axon, down to the next neuron, and then they're also going to be able to secrete things. When that electrical signal gets to the end, it needs to be passed on to the next neuron. And we've seen pictures of synapses, right? There's chemical messengers that are going to be secreted from vesicles into the synapse to be received by the next neuron, which activates the next electrical signal to be passed on to the next neuron. We have a few different types of neurons. We have sensory or afferent neurons. These are just the neurons that are going to take signals from the outside of our body and send them inwards towards the central nervous system. There are interneurons. These are going to be within the central nervous system. So the signals will come from the outside, go into the central nervous system, but the central nervous system, we said, is where the decisions are going to get made. And so different neurons are going to talk to each other to try to figure out what the best course of action is. And so that talking from one to another is going to happen through interneurons. And then, once the decision is made, and the signal needs to go back out to the muscles, the glands, and the organs, that's going to happen through a motor or efferent neuron. So this is what we're talking about. So we have the sensory neurons are going to feel something. That signal will go into the central nervous system. The central nervous system will make some phone calls amongst each other, passing around responsibility for making a decision. Finally, the boss will make a decision and then tell the workers out in the real world what to do, right? And so we have sensory, interneurons, and motor neurons. This is essentially where we started talking in the lab. And so these are the parts of a neuron. The main body of the cell, remember a neuron is a cell. It is one single cell. The main part of the cell is the soma, also called the neurosoma or cell body. And it's going to be the large round area that includes the nucleus. Neurons are not mitotic, which means they are not dividing. Other cells will divide to make neurons. But once a cell is a neuron, it will not divide and reproduce anymore. Remember that the dendrites are these small little branches coming off of the cell body. These are going to receive signals from other neurons. Messages are received by the dendrite, and then they'll move down the dendrite into the cell body. The axon is the very long fiber that comes off of the cell body. Remember that, and where this kind of tapers down, that's called our axon hillock. Because we're going to have more or less one long strand. We might have one or two coming off the side, but until we get to the end, it's more or less one process. And then at the very end, we're going to have a whole bunch of different fingers, because then these little fingers are going to mesh with the little fingers on the dendrites of the next neuron. Question?
So any of those branch offs would be just considered dendrites?
Say it again?
Any of the branch offs would be considered dendrites?
Up here.
Right off to the left.
Here?
No, the long one up towards the left. On the axon.
Here?
Lower, right there.
This?
Oh,
So this?
yeah, that right there.
So this is just a branch of the axon.
Okay, so what would that be called?
It's just axon.
Okay.
It's just axon. And so most general diagrams that we have only have one, but it can branch, but it's still just an axon. It's going to act exactly the same. It's just that signal is going to branch and go to two different places. Okay. at the very end, where it branches, we have synaptic knobs. So this is going to be what goes into the synapse. The synapse, remember, is the little gap between two neurons. So we have the axon coming down from the top, then we have a little gap called the synapse, and then we have a dendrite from the bottom. And so we'll see, there'll be a picture, we'll have the same diagram again. We'll see the synapse, the very end of the axon is going to be kind of a bulb shape. It's not just going to come down in a tube and get cut off. That bulb is going to be called the synaptic knob, and that's where our vesicles containing our chemical transmitter are going to be. Neurons, like we saw, can have different shapes. They're not all the one neat axon that we like to put in our diagrams, in our models. And so this is a multipolar neuron. And so we have dendrites, but we have multiple axons, right? So here we have two axons coming off straight from the cell body. Here is like what we just had in that picture. So we have one that comes off, that comes down, but then there's another one coming off the side. This is a multipolar neuron. Yeah.
How do you know which ones are which?
In which way?
The
Like-
axon and the dendrites.
Yeah, how do you know
I
which ones?
see.
Do you know what I mean? They look the same. I mean, if you didn't point them out, I wouldn't know which one's which.
Unless there's no branches with the axon. Well, there is a little.
Well, the axon's gonna have the myelin sheath, right?
Yeah. I think they probably don't do them.
Maybe that's just a bad example.
I would normally imagine the axon here being wider and thicker than the
Than the-
dendrite.
yeah.
would be here.
Okay.
The way they have it drawn, they don't really have them drawn different sizes.
Okay.
I would expect the axon to be thicker than this.
Okay. Okay.
So that's a multipolar. A bipolar neuron is going to be what we pictured originally with a neuron, with one set of dendrites and one axon coming down. It's bipolar because there's just two ends to it, a dendrite end and an axon end. But we can also have unipolar neurons. So this is a unipolar neuron. And so there's just going to be one tube coming off of our cell body. And actually, now that I look at it, I don't like. I don't like this picture. Why, can somebody tell me why I don't like this picture? I'm just now realizing. It has two. It has two. So based on the description there, what would you expect it to look like? Just one. And what would that one thing be? Would it be dendrites or an axon? An axon, right? The purpose of the dendrites is to receive signals and then pass it on. But if you're the first one in line, there's no one to receive a signal from. You are the security guard watching for something to happen, right? You're sensing the initial stimulus. And so you sense it yourself, and then you send that signal out.
So
That
that
is
should
not
have
the-
dendrites on
No, 2 axons.
it? Or dendrites at the end?
Yeah. So, I don't know. They have axon. They have 2 axons here, which I love, which is okay. But down here, they have it labeled dendrites. Those aren't dendrites, right?
Right.
Dendrites are not at the end of an axon. So imagine that being gone, that label being gone, and these are just 2 axons. Now it makes sense. Does everybody see that? So if you have your slides, just cross this out. And so this cell body is some sort of a sensor, whether it be temperature sensor or a pressure sensor or a touch sensor. Something's going to happen to that neuron. And then it's going to initiate the signal based on that sense and send it down the axons to multiple second neurons. Good?
You said that the axon shouldn't have dendrites, but what about the other side? Isn't that dendrites on the right side?
Here?
Terminal arterization.
Yeah, they're not dendrites.
They're
So
not.
they only labeled one end. These two ends should be the same thing, but they labeled one end dendrites, and they're not dendrites. Because the signal is going down the axon to that end. So it's going to send it to the next neuron. That's not a dendrite.
Okay.
It has, if you Google this, like for the lab, for the final, they have a really good picture online where it's already labeled and it's the actual model from our lab class.
Really?
Yeah, I mean, it's labeled, because it's kind of confusing, because one of the models is kind of weird in lab. So I
Just
would
one?
look that up too. Well, it has it all labeled of which one's dendrite and which one's the axon, because it kind of is weird. Just saying.
So everybody Google it, right? That's the answer to everything. Google it. So the fourth type is an anoxic neuron. So this is going to be only dendrites. This is axons, no dendrites. This is dendrites only. Why would you only have dendrites only? Right. If you are the end of the line. So this is the start of the line. This is the end of the line. These are going to carry a signal between the two. So this is going to be in your central nervous system, like your brain, and it's going to receive signals. It's going to receive a signal, and then it's going to do something with that signal. It's not going to just pass it on to another neuron. Okay, so that's the neuron itself. Then we have the neuroglia, which we've talked about. So remember that neuroglia are actually by far the most common cell in the nervous system. We have far more cells to support the neurons than we actually have neurons. And they serve the purpose only of helping the neurons do what they need to do. In the central nervous system, we had 4 types of neuroglia. There were oligodendrocytes, ependymal cells, microglia, and astrocytes. The oligodendrocytes, remember, were very similar to the Schwann cells. Schwann cells are in the peripheral nervous system. Oligodendrocytes are in the central nervous system. They're going to put the myelin sheath on the axons. But rather than being like a Schwann cell that's just wrapped around an axon, it's going to be kind of like an octopus with a bunch of tentacles coming out, attaching to multiple axons and wrapping around them. The ependymal cells are located in the brain and the spinal cord. They're going to make the cerebrospinal fluid. The microglia are sort of like the macrophages in our skin. They're going to look for debris and invaders in our nervous system. Then there were the astrocytes. Astrocytes are by far the most common type, and they have a whole bunch of different purposes. So they're going to kind of form the skeleton of the nervous system. They're going to facilitate movement of blood from the capillaries and blood vessels around the nervous system to the neurons. They're going to be kind of almost like digesting the neurons' food for them. So the neurons don't even have to focus on metabolism. They're going to secrete growth factors. They can communicate directly with the neurons. They're going to control the environment around the neurons, keeping all the ion concentrations correct so that the neuron will work correctly. And then they're actually going to step in if a neuron is damaged and kind of act as a scab. So what we're going to see, just like with our muscle, it's all about flow of ions and electrical charges. If you punch a hole in a neuron, the ions are just going to flood in and out, and you're not going to be able to create that voltage difference. So the astrocyte will just kind of go on as a patch. Yes, you lose function of that spot on your neuron, but at least you're not leaking in and out, so the rest of it will be able to work. So this is our diagram that we saw before with our neuroglia. So this is our oligodendrocyte wrapped onto multiple axons. In the peripheral nervous system, there are only two types of neuroglia. We have the Schwann cells, which we're all very, very familiar with at this point. And then there were satellite cells. So these are going to be around the neurosoma. What was the neurosoma? The cell body. That's the main part of our neuron. And so these are going to serve a similar purpose to the Schwann cells. Remember, the myelin is sort of the insulation. It's going to prevent the electrical signal from leaking out of our axon. But we have that gigantic cell body up there with no myelin on it. And so these satellite cells are going to kind of act as a cross between a Schwann cell, providing the insulation, but it's also going to act kind of like an astrocyte, and then in regulating the chemical environment around our neuron. And so the peripheral nervous system isn't as complex as the central nervous system. It doesn't have as many jobs that it needs to do, and so it's able to do all of those jobs with just the Schwann cells and the satellite cells helping the neuron. Myelin, we're good with. Schwann cells. Dendrocyte, myelin. Okay. So this is the movement of those electrical currents. This is something we haven't talked about in terms of the nervous system yet. But you're going to see it's fairly similar to what we just talked about in our muscle tissue. We're going to see ions flowing in and out of a cell. That's going to cause changes in voltage, which are going to trigger larger changes in voltage. The difference is going to be what then happens because of that. In the muscle, that caused the release of the calcium, the muscle contracted, and that was the end of it. Here, we're going to be sending that signal down an axon, through a synapse, into a dendrite, into the next cell body, down the next axon, and it's going to be repeated over and over and over again. So remember that an electrical potential is voltage. It's the difference in voltage between one side of the membrane and the other side. So it's the difference in charge inside of a cell versus the outside of a cell. All of our living cells are polarized, meaning there is a difference in voltage from the inside of the cell to the outside of the cell. And that creates what's called the resting membrane potential, which we saw in our muscle cells, right? If it's just sitting there, not activated, there's this charge difference that's waiting to be activated and kicked into gear to cause ions to flow in. A cell, including nervous system cell, has more negative particles inside than outside, which means that when you have a cell membrane on your neuron, this is the inside, I just draw a circle, and that has a clear inside. Circle. So there's the inside. The inside of your cell is going to be negatively charged. Outside will be positive. And so the resting membrane potential for a neuron is about negative 70 millivolts. So that tells you the scale of the difference between the negative inside and the positive outside. If we had something like negative 5 millivolts, that would mean that the difference between the inside and the outside is smaller. So the absolute value of that number will tell you how large the difference is. Whether it's negative or positive will tell you which side the negative is and which side the positive is. So here, the negative is on the inside. Electrical current is going to be the flow of electrically charged particles. And the outlets in the wall, those are electrons that are going to flow. Here we're not dealing with electrons, we're dealing with ions. So these are atoms, not electrons, but entire atoms that have a charge to them. So they are going to flow in and out of our cell, and they're going to want to get rid of this difference. Charges don't like to exist. The negatives in here want to come out. The positives on the outside want to come in. They want to make everything equal. And so, luckily, our plasma membrane prevents that from happening. But when we give it a chance, by opening a pore, things are going to move in and out as fast as possible to try to equalize that. We'll use that to our advantage, we'll let that happen, and then we'll shut it off, pump everything back out, and do it again. So remember, extracellular fluid is going to be essentially the water outside of our cell. That's going to be our positively charged area. The intracellular fluid will be inside of our cell. So we have ECF and ICF. So this resting membrane potential is going to be set up by a few different factors. Number one, remember, there's going to be two things that are going to be different on the inside and the outside. There's charge difference, right? And so the positives outside are going to want to come in, and negatives on the inside are going to want to go out. But at the same time, we are going to have osmosis happening. We're going to have things that are positively charged on the inside that are very, very concentrated that want to go out. So the fact that they are positive says that they want to come in, but the fact that they specifically are concentrated on the inside will make them want to go out. And it's going to be a balancing act of what that atom is going to find more important at that specific time. The fact that they're positively charged, or the fact that they're a potassium. And potassium wants to go out. It is the catch-22. It's going to be which difference is larger. Is the voltage difference larger, or is the concentration difference larger? Whichever one is larger is going to win. So this ions diffusing down their concentration gradient is going to be things like potassium, which we'll see is very concentrated in here, is going to diffuse out where it's not concentrated. That's our osmosis, right? But at the same time, the electrical attraction of the positive and negative is going to want to pull positive potassiums that are out here into the cell towards the negative, and the plasma membrane is going to prevent these things from happening automatically and control when they can and can't happen. So potassium is the key here. In the muscles, we had sodium coming in, and that was the key, releasing calcium, which was the key to activating the muscle. Here, potassium is going to be the most important ion. Where the potassium goes is going to determine if the neuron is active or not. The plasma membrane is actually somewhat permeable to potassium. Potassium can just leak through a little bit. Most other ions and molecules can't. It's like a wall, it can't get through. But potassium can leak through a little bit. And so it's very concentrated in here. But because it's concentrated in here, it wants to leak out. So if I have a whole bunch of potassiums in here, this one is going to say, well, there's 4 potassiums inside, none outside. I want to go outside to get away from the rest. So he is going to come out. But when that happens, I remove a positive from inside the cell. Now then this one leaves, and that one leaves, and that one leaves. The potassiums are going where potassium wants to go, but they've lost sight of the bigger picture. Because they're leaving, now we don't have enough positives inside. And so now the negatives outweigh the positives, which is where this negative charge in our cell initially comes from. We pump a whole bunch of positive potassiums in, and then when they leave, they take the positive charge with them. And that leaves behind a negative. And so it's not like we're pumping negatives in, we're allowing positives out. Does that make sense? Because in a neutral situation, you would have an equal number. If you have 50 of one and 50 of the other, you're neutral. But if 5 of your positives leave, now you have 50 negatives and 45 positives, which is a net of 5 negatives. You have 5 more negatives than positives. And so this is saying potassium is 40 times more concentrated inside than outside. So that is a very strong force. That force is actually stronger than this charge difference. And so potassium will just leak out on its own. The negative ions can't get out. Things like chloride from sodium chloride are stuck in here. They can't just leave in order to balance it out. The only thing that can just get through the plasma membrane are the positive potassiums. Sodium is also going to have some effect on our membrane potential, but not nearly as large as our potassium, because sodium can't get through the plasma membrane nearly as well. We have about 12 times the concentration of sodium outside the cell as inside. So this is a very important point. Potassium is concentrated inside the cell. Sodium is concentrated outside of the cell. That's a very, very important concept. So because sodium is so much more concentrated on the outside than the inside, some of it is going to leak in. The concentration difference for sodium is not as large as it is for potassium, and sodium can't get through the membrane as well. And so it leaking in happens much slower than the potassium leaking out. But it does happen, and so it does have an effect on the resting membrane potential. Where things can really speed up is with the sodium-potassium pumps, which we saw in probably our cell lecture, right? So we can take 3 sodiums, pump 3 sodiums out for every 2 potassiums that we pump in. And so because of that, we pump 3 positives out. For 1 turn of the crank, we pump 3 positives out and 2 in. So in terms of just electrical charge, we're causing an increase in that charge difference. We're putting 3 positives out, but only bringing 2 in. And so the number of positives inside of our cell is going to decrease and decrease and decrease. Because this is active transport. This requires ATP. If you require ATP, you need glucose and oxygen. And so if we were to lose oxygen to our body, say when we die, we're going to lose the ATP in our body, and this is going to stop happening. Resting membrane potential will eventually go away. This requires the use of energy. So this is what we're just talking about. So here we have our plasma membrane, our cell membrane, and we're going to have very high concentrations of sodium outside compared to the inside, and we'll have high concentrations of potassium on the inside compared to the outside. The numbers aren't important to you. I just want you to see that the sodium is higher outside than inside, and potassium is higher on the inside. And so sodium there are channels for sodium and potassium, but they can also leak straight across the membrane. Where the channels are going to come in is when we actively turn this neuron on. When we turn a neuron on, we're going to create what's called an action potential. But there are also local potentials. Potential is going to be a difference in charge. So imagine I have a cell starting out. And I have my resting membrane potential. And that resting membrane potential is negative 70 millivolts. If I open a sodium channel here, so I am going to let sodium move through that channel, which way is it going to go? Just based on the concentration of sodium. Which way? Inside. We're starting with a whole bunch of sodium outside and very little in. So when you open that channel, sodium is going to rush in. When that happens, you're going to end up with a whole bunch of sodium right at the inside of that channel. It's going to take some time for this to diffuse throughout the cell. And so, for this part of the cell, it's still basically negative 70 millivolts. But in this little area where the sodiums have come in, it's not negative 70 millivolts anymore. Now maybe it's negative 50 millivolts. Positives came in, and so my charge is going to get less negative. That's called a local potential, because the charge difference is only localized. It's in one part of the neuron. This is what's going to activate an action potential. Think about in our muscles. We talked about when that signal comes, sodium's going to come in one pore, which is going to activate voltage-gated channels, which is going to let a whole bunch in. It's the same idea. We're going to have sodium coming in one channel here, creating a local potential, which is going to cause channels over the entire surface to open up and let a flood of sodium come in. So this channel, the initial channel, can be activated by a number of different things. Chemicals, whether it's a synapse like we've seen, light in the retina of our eye. These cells actually get hit by photons of light, causing these channels to open up. Temperature sensors in our hands and our fingers work the same way. The temperature increases, these pores open up. And so the higher the temperature, the more sodium comes in, going to create a local potential, and also mechanical disturbance. If I touch the board, the physical touching of the board is going to cause some of these channels to open up. So local potentials are localized, they're small area, but the way they behave is going to be different than our action potential. Number one, they are graded, which means the stronger the stimulus, the stronger this potential is going to be. If I hit this cell with a very strong stimulus, more of these pores are going to open up. And the more pores that open up, the more sodium will come in, and the larger the charge difference will be. They're called decremental, meaning that the further away you get from the open pore, the closer you'll get to your resting membrane potential. So it's not that there's this little pocket that's negative 50. Maybe right next to the pore, we're at negative 20. And then here we're at negative 30. And then here we're at negative 50 and negative 70. So there's a change in voltage as we get away from the open pore. Action potential is going to be different. everything is going to be the same charge. It's also reversible. As soon as the stimulus is gone, that pore will close and the whole thing will shut off. Sodium will get pumped back out and you'll go right back to where you were. An action potential is going to be like a chain reaction. Once you start it, there's no going back. You have to let it finish its course and then you can stop the next action potential. Local potentials can be excitatory or inhibitory. So what this is, with sodium coming in, is excitatory. Positive ions coming into the cell are going to turn an axon on. But if instead of a sodium channel, we opened up a chloride channel. And so we're going to have chloride coming in. This is going to make it more negative. What we're going to see is that the action potential in the neuron signal is caused by positive changes. We have to go towards 0, from -70 up towards 0. If we let in more negatives, we're going away. And so in order to overcome that, we're going to have to let in even more sodium. And so allowing negatives in would be an inhibitory process. So here we have this happen. So we have a signal that we're being received in the dendrite, and we're going to cause a local potential. That local potential is going to travel down the dendrite into the soma and hit the trigger zone. If when it hits the trigger zone, the voltage difference is still high enough, it's going to be above that threshold voltage, then the axon is going to fire. So we're going to have a slow, weak signal coming through the dendrites, through the soma, to the trigger zone at the axon hillock. If it's still strong enough there, then it's sort of all hands on deck and fire all the cannons at once. And then the signal will go down the axon, down to the next dendrite. And so here, this is in our dendrite. So we have our plasma membrane with a bunch of sodium on the outside, a little bit of sodium on the inside. This dendrite just received a signal from another axon that gave it a chemical messenger from the synapse above it. That chemical messenger is going to bind to this channel, cause it to open. When it opens, sodium is just going to pour in. That sodium will then slowly work its way down through the dendrites, through the soma, to the trigger zone. If when it hits that trigger zone, we're going to create this action potential. And this is a very dramatic change in voltage. It's going to be very large, and it's going to be very, very quickly. And this is going to be based on voltage-gated ion channels. The voltage-gated ion channels are located in that target zone. If all of a sudden the area inside the cell there becomes not -70, maybe it's -50, then they're going to open, a whole bunch of sodium is going to come in, and we're going to cause this action potential. In order for the action potential to happen, we need a very high density of voltage gates, voltage regulated gates. We have to have a lot of them. We need a lot of sodium to come in very, very quickly. If we don't have enough of these voltage gated channels in that area, you can't create an action potential. On the soma, the cell body, there's only 50 to 75 per square micrometer. So that's not really a unit you're comfortable with. But it's imagine a small little area, and in that area, there are 50 to 75 of them. But in that trigger zone, in the same amount of area, there's 350 to 500 of them. So up here in the soma, you're just kind of helping that local potential move through the cell body. You're just kind of giving it a little push in the behind, helping it get through. But the trigger zone is where everything's going to flood in and cause the chain reaction to happen. So action potential is going to initially go up in voltage. We're starting negative, we're going to allow a whole bunch of positives in. So the voltage is going to spike up into the positive range, but then it's very quickly going to crash back down. And so that current change is going to arrive at the axon hillock, the target zone. If it reaches threshold voltage, which is about -55, that number is not important to you. You just have to know the concept of threshold voltage. The voltage regulated gates will open, sodium is going to rush in, and that's going to cause even more voltage gated channels to open. Because when some open, the voltage gets stronger, which causes more gates to open, which makes the voltage stronger. And a third batch is going to open. And so the voltage is going to start at -70. So we're starting at -70, and then it's going to go to -60 and -50, and eventually it's going to hit 0. When it hits 0, the voltage-gated sodium channels are going to close. You're going to say, no more. I can't allow any more sodium in. But you can't close it fast enough. And so some more is still going to go in, and so it's going to kind of peak at +35. So in the blink of an eye, we went from -70 to +35. So on a graph, we'll see in a graph, it just kind of spikes up, and that's going to come back down. So up here, we're at about +35. We started at -70. When we hit here at 0, the sodium is going to close, but the potassium channels are going to open. Potassium is going to want to leave the cell. Potassium is positively charged. And so as the potassium leaves, it's going to bring us back towards -70. So potassium leaking out is going to bring us that way. Sodium leaking in is going to take us towards the positive. No, so it's leaking out. And so it's not adding a negative, it's removing a positive. So this is our graph here. So we're down here at the resting membrane potential. Some sort of signal comes in, it opens the initial sodium gates. We create a local potential that gets stronger and stronger and stronger until we hit threshold. When we hit threshold, all those voltage gated channels are going to slam open. Our voltage is going to spike up to +35, but then the sodium channels close, potassium channels stay open, so the positives leak out, and it comes back down. This process here of coming back down is called repolarization. So polar is saying it's different inside and outside. Repolarization is getting back to that state. But you notice when it comes down, it actually briefly comes down below where we started at -70. Say it again?
That's how it'll start again?
That's actually so it won't start again. So at this period down here, we are below -70. So over here, we talked about if you make it more negative, it's harder to activate. And so for a brief period of time, we're going to be what's called hyperpolarized. We're down below -70. The purpose is to try to keep this from firing too quickly, one after another, after another, after another. It's going to try to force the cell to give it a little bit of recovery time in between action potentials. It's going to space those signals out for us. So this is what we just talked about. So this is kind of resting. Both channels are closed. Sodium's going to open. Sodium rushes in, sodium closes, potassium opens, potassium comes out. And so now we're going to come back down. Here we're at rusting, it's -70. Sodium opens, we come up, we hit the peak at the top, sodium closes, potassium opens, potassium leaks out, and our graph comes back down to the bottom. So that hyperpolarized part down here is part of what's called the refractory period. So there are two parts of the refractory period. The absolute refractory period and the relative refractory period. In the absolute refractory period, the cell cannot send out another action potential. It just cannot. No matter what you do, it cannot do it. In the relative refractory period, it can, it just doesn't want to. And so you're going to have to give it a stronger stimulus than you did the first time in order to get it to go. Yeah?
What would that look like physically?
What would it look like physically?
What could we not do that would enable us to do that? You know what I'm saying?
No, I don't think I do.
They're out there, wow.
Okay. So this absolute refractory period is going to be when those sodium gates are open. They're open. They can't do any more. You can't send the second action potential if you're already just doing the first one. The relative refractory period is going to be when the potassium gates are open. And so we're still bringing our charge down. We're low enough that we can fire again, but potassium is still open. And so the relative refractory period is going to end once we stop our hyperpolarization and we come back up to -7. So as this action potential moves down an axon, there's two ways that it can move. And it's going to depend on whether that axon has myelin on it or not. An unmyelinated nerve fiber is going to have voltage-gated channels along the entire thing. And so as the charge moves down the axon, new voltage-gated channels are going to open as it goes. It continues the signal down the string, down the axon. So here we see this is an axon. And so the red is our action potential moving down an axon. So here is where the electrical signal is. It's moving this way. Because of that refractory period, as it moves down, part of the axon where we just passed is refractory, which means that the signal can't reverse and go back that way. The only way the signal can move is this direction because this is not refractory. This is refractory, so it can't turn around and go back that way. Myelinated fibers move differently. This is called saltatory conduction. So remember what our myelinated nerve fiber looks like. There was the Schwann cells and oligodendrocytes with the myelin, and then there were the nodes of Ranvier. Remember the little spaces between the myelin. In a myelinated nerve fiber, the voltage-gated channels are only going to be located at those nodes of Ranvier. And so what's going to happen is because the myelin is acting like insulation, none of this charge can leak out. So it's going to come in one node, it can only go one direction, and none of it can leak out. So it's very strongly and very quickly going to move down the axon to the next node, where there's a whole bunch of channels waiting. So when the signal gets there, they hit it hard again. And the next one hits it hard again and again and again. Because of that, it moves much quicker. So you're going to see it's going to move down, it's going to hit one node, a bunch of it's going to move into the axon, a bunch of sodium is going to come into the axon, it's going to move past the next myelin sheath to the next node, it's gonna punch it again. So I have